It's 6:38 p.m. on a Wednesday night. You just finished a long day of work. You unlock the door, ready to have a restful night. But instead, there you lay, thinking about the future. But not your future, your child's future. Will they have diabetes like your grandma? Are they prone to having eczema like your brother? And will they eventually develop dementia like your uncle did? You watch the people around you wither with age and struggle with many disorders. But what if your child's life is different? What if you can change their genes and ensure that their life will be better? One of the ways we can customize babies is through embryonic genome editing, but this requires 3 main steps. The first step is when fertilization of the egg occurs inside a plate, known as in vitro fertilization. This is followed by a preconceptional genetic test, which is a biopsy for the embryo that acts as a DNA scan to understand what hereditary diseases the future child might have, allowing the parents to choose what illnesses they want to focus on and eliminate. The final step of the process is the insertion of CRISPR gene editing technology into the embryo's pronucleus, where the DNA is found. This step must happen in the early stages of embryogenesis, such as the zygotic stage, so that once the cell starts splitting and replicating its DNA, it can replicate the edited genome. Although this is not currently in practice on humans, it has been in use on mice as early as 1974, where scientists inserted a virus into a mouse's embryo and found that the viral DNA was integrated into the mouse's genome. This experiment showed that with careful techniques, humans have the ability to insert new genes into an embryo and have them become part of the genome being targeted.
CRISPR is a revolutionary gene editing tool that allows scientists to precisely alter DNA sequences in living organisms. It works like molecular scissors guided by RNA to target specific genes and either cut them out, modify them, or replace them. Originally discovered in bacteria as a defense mechanism against viruses, CRISPR is now used in medicine, agriculture, and research to address challenges like genetic diseases, crop improvements, and even creating treatments for conditions like cancer. Its accuracy, efficiency, and versatility have made it one of the most powerful tools in modern science.
Now we'll be moving on to germline editing. So what is the difference between embryonic editing and germline editing? In humans, we have specialized reproductive cells. They're called sperm in males and eggs, or ovum in females. Germline editing refers to the editing of these cells, so either before the fertilization of the egg, when the sperm meets the ovum, or very early on after fertilization, right before it becomes an embryo at 8 weeks. This means that we often do not know if the embryo is going to have a genetic defect. It is more targeted to generationally inherited diseases instead of observed defects like in embryonic editing. Furthermore, this also means that germline editing is more likely used for CRISPR-induced enhancements, such as specific facial features, longer limbs, more height, etc. This also means that germline editing carries its own ethical concerns of whether consent exists within the space or if this causes a competitive advantage and enhancement to those who have access to these possible procedures. Due to these encounters, much research has gone into using germline editing to study disease, where disease can be induced on animal models such as lab mice to study the effects of treatment. Think of a world where everyone around you has a enhancement because of germline editing.
The implementation of CRISPR technology in germline and embryonic cell editing has profound implications for the field of genetic engineering. It represents a groundbreaking shift, allowing scientists to target and alter DNA in ways that can permanently impact future generations. This opens up possibilities for eradicating genetic diseases and selecting desirable traits, but it also introduces complex ethical, societal, and equity challenges. By enabling such precise genetic manipulation, CRISPR pushes the boundaries of what humanity can achieve, while simultaneously raising questions about fairness, access, and unintended consequences. The ability to customize traits in future offspring blurs the lines between medical necessity and enhancement, sparking debates about the potential for designer babies and social inequality. Overall, this technology is shaping the field by merging scientific innovation with ethical reflection, forcing researchers and society to navigate uncharted territory in genetic science.
But what does all of this really mean for the child's health? CRISPR could be transformative for a child, and the research has already begun. Lulu and Nina were the world's first genetically engineered babies with CRISPR, and they were created to be resistant to specific disorders like smallpox, HIV, and cholera. While the potential is groundbreaking, the risks are still significant. CRISPR is not perfect. We could intend to edit a gene with CRISPR, and something else could end up happening. Kind of like how Patrick tries to fix SpongeBob Splinter with a hammer and it ends up backfiring. Off target effects or editing errors can lead to unintended mutations that can have negative effects on a child's health. Because CRISPR is new and limited research has been done on humans, the nuances of this are also widely misunderstood. Accessibility to CRISPR could also widen social divides, with wealthier families more likely to afford genetic enhancements. This raises the question: will society be divided between the genetically modified and the naturally born? Regulation is also another challenge. In Canada, the Assisted Human Reproduction Act strictly prohibits genetic editing of embryos, making the pathway to clinical application uncertain.